Good afternoon, everyone. We have hit the top of the hour, so I am going to go ahead and get started. So good afternoon, and thank you for joining us for this Board of Animal Health webinar. My name is Dr. Caitlin Macy, and I am a district field veterinarian with the Board of Animal Health. I will be the moderator for today's webinar. Today, we have three speakers that will be presenting on highly pathogenic avian influenza's impact on wild birds and on human health. Just a couple of housekeeping items before I introduce our first speaker. We will have a question and answer session at the end of today's talk. You are muted. If you think of questions during this presentation, either the first one or the second, please go ahead and type them into the chat box, and we will try to address as many as we can. You can direct them either to the Board of Animal Health or everyone in the chat section. As we know from last week's webinar, there will likely be many more questions than we can answer in our allotted time. So any questions that have not been answered by the end of this webinar will be compiled, answered, and attached to the YouTube posting in a week or so following this webinar. You are able to receive continuing education credit for this webinar. If you have signed in on Teams with your name, we will send you your certificate to your email address. If you called in, please email us to let us know that you joined, and we will go ahead and send you your certificate. And with that, I will go ahead and introduce our first speaker. Our first speaker is Eli Fleace. Eli Fleace is a fish and wildlife health biologist with the Indiana Department of Natural Resources and is the avian health specialist. He graduated from Ball State University in 2020 with a bachelor's in biology with concentrations in zoology, wildlife biology, and conservation, and botany. Eli has been organizing the DNR response to highly pathogenic avian influenza. And with that, I would like to welcome you to begin, Eli.
Hi, thank you for the wonderful introduction. Like she said, my name is Eli. I'm a avian health. Oh, can you not hear me?
I can hear him.
Perfect.
Okay. Can you hear me now?
Yes.
Okay. So thank you for the introduction. My name's Eli. Like she said, I'm a fish and wildlife health biologist with the DNR and the avian health biologist. I threw this presentation together last night, so forgive me if it's not the best presentation, but things have been pretty busy. And I wanted to try and keep it short so there'd be more time for questions. So in the spirit of keeping it short, I guess we should go ahead and jump straight in. So highly pathogenic avian influenza in wild birds. I know there's a lot of questions about that, and it seems very relevant at the moment with everything that's going on across the landscape of Indiana. Would you go ahead and give me the next slide, please? So I wanted to just give like a quick background on avian influenza. I know most of the people here are probably very familiar with it. It's an influenza type A virus. It's a RNA virus, and there are two types of avian influenza that we break it down into two different types. There's highly pathogenic avian influenza and low pathogenic avian influenza. And those terms are related directly to domestic poultry. So highly pathogenic avian influenza is bad for chickens. It's really pathogenic for domestic poultry. It has really negative effects for them, whereas low pathogenic, it doesn't really affect domestic poultry nearly at the same level as highly pathogenic. And so those terms don't really relate to effects on wildlife health or human health. You can have a highly pathogenic strain of avian influenza that doesn't really affect wildlife, but it could affect domestic poultry. I also want to say that low pathogenic avian influenza, it's really common to find that in wild duck species. They pretty much carry low pathogenic strains of avian influenza throughout most of the year. We do some testing on live captured ducks during the summer, and most of the virus that we have detected have been low pathogenic strains. And so these ducks just kind of carry it with them. They don't really get sick from it. They just have it in their system pretty much at all times. And you can really see that affect their immune response. Typically, when we see outbreaks of avian influenza, highly pathogenic avian influenza, the ducks aren't quite as seriously affected as the geese are, typically speaking. So it seems like the ducks tend to have a better immunity to it, and that's probably because they just carry those low pathogenic strains with them at all times. Next slide. So avian influenza is further broken up into different types, and those types are based on two surface proteins that the virus has. There's hemagglutinin and neuraminidase. There are 16 types of hemagglutinin and 9 types of neuraminidase. Technically, there are 18 types of hemagglutinin and 11 types of neuraminidase, but type 17 and type 18, H17 and H18 and N11 and N10 have only ever been detected in bats in South America. So nobody really talks about those two, those types. But functionally, there are 16 types of hemagglutinin and 9 types of neuraminidase. And I wanted to point out that these two surface proteins, the reason that they're important, the hemagglutinin kind of determines how the virus binds to host cells, and the neuraminidase kind of determines how the virus detaches from host cells to spread out further into the body. And so depending on the chemistry of the host cells, these different types can have different effects on other species. So there's quite a bit of variation in how the virus affects different species just based on the typing of those surface proteins. Next slide, please. So the strain that everybody's concerned about, the one that's really affecting North America and much of the world at the moment, is H5N1. A little background on H5N1, it's been around for a while. It first appeared in China in 1996, which is the year I was born, so this virus has been around as long as I have. And there were initial outbreaks in some poultry in China. I think the current theory is that there were some ducks that had some interaction with some domestic geese, I believe, in China. And that kind of created this strain of H5N1. But it was really localized to this specific region in China and Hong Kong for a few years. And it really didn't start to spread until about 2003. Where it started to spread out across China and then a couple other different countries in Asia. But then by 2006, it really started to take off. It had spread to 53 countries, and it was making its way into Europe and Africa. And it was pretty much across that whole Eurasian supercontinent. In 2014, a new genetic lineage appeared, the 2.3.4.4 lineage. And that really became the source for a lot of waterfowl and wild bird deaths. Whenever they would type out a strain of avian influenza that had caused a large mortality event, it was usually in that genetic lineage. And so it circulated through Europe and Asia for about 7 years. And then in 2021, it was picked up by some wild migrating birds and brought across the Atlantic up to North America. I think it was first detected in North America in 2021, December of 2021 in Newfoundland. And after it got introduced in Newfoundland, it really quickly started to make its way south on the eastern border of the U.S. And then across the inland. And then it spread across the American supercontinent very fast. By 2023, it had already been detected in Argentina. So less than 2 years, it had made it from the far north of the Americas all the way to the bottom of the Americas. Next slide, please. So bringing it a little closer to home, I wanted to kind of explain some of the ways that avian influenza spreads in these wild birds. As it relates to wildlife, it's typically a cold weather virus. And there are several reasons for that. Part of that is migration patterns. So a lot of these birds are migrating in the winter. And so they're moving across huge tracts of lands from different spot to different spot. And a lot of times they're interacting with a lot of other migrating birds, and that just creates a lot of opportunity for the virus to spread. There's also some flocking patterns. While they're doing these migrations, they're flocking in huge numbers. Depending on the species, like in snow geese, we see flocks of, tens to hundreds of thousands of birds all grouped together. Sandhill cranes, we see them flock together and by the thousands and thousands. And so when these huge groups are all grouped together, they're all landing in similar water sources nearby. They're sharing food sources. They have really high interaction with each other. And that's just makes for a great opportunity for the virus to spread. I put finding food on here because that is a good illustration of how the virus can spread across species really easily. During the winter, it's a little more difficult to find food than it is in the summer for wildlife. And so if there is an available food source, you'll see multiple different species, multiple different taxa, all grouped together in those areas. And they're, walking through each other's feces, they're breathing on each other, they're eating the same food. There's really high interaction, and that creates a great opportunity for the virus to spread between species. And the virus itself, maybe my public health colleagues can speak to this a little better than I can, but I believe the virus itself, just the way that it's structured chemically, it survives in the cold better. I think it's because there's a lipid barrier on the outside of the virus that hardens in cold weather. And so the virus itself can stay sustained in cold weather longer than it can in warm weather. And I didn't type this one out, but the virus can also move through the air in cold weather a lot more easily. There's a lot less humidity in colder weather, which is why my face always gets really dry skin in the winter. But the virus can, because there's not as much humidity, the virus can move through the air a little bit easier and stay suspended in the air for longer periods of time. Next slide, please. This virus primarily affects waterfowl traditionally, and that's ducks, geese, and swans. Like I said earlier, the ducks tend to have a little better immunity to it, so we don't see quite as dramatic mortality events in the ducks as we do in other species. But it does cause some deaths in the ducks. There was several dozen mergansers that died up in Lake Michigan to it. So the ducks aren't fully safe from this virus, but it does seem to have a lesser effect on them. The virus also affects other species, raptors notably, and some marsh birds and shorebirds. Sandhill cranes are a big one at the moment. There's a lot of sandhill crane deaths across the state due to avian influenza. But it does affect quite a few species across a lot of different taxa. And it's also, in the last few years, it's been detected in an increasing number of mammals as well. In Indiana, that's kind of relevant because it's been found in raccoons and foxes and possums, but outside of Indiana, it's been found in all sorts of stuff like dolphins and sea lions and bears and all sorts of stuff. And I know nobody on this call really cares about domestic animals, but it can also be, it can affect domestic goats, dairy cattle, and especially house cats. House cats have a really, really tough time with this virus. And obviously domestic poultry as well. Next slide. So a quick timeline of HPAI in Indiana. I'm going to start in 2022 when H5N1 was first detected. I believe it was actually detected in domestic poultry before we found it in wildlife. I think the first detection was in a turkey farm in February of 2022. And then in March, we detected it in a red-headed duck. And through March to early summer, we had a scattering of reports. A lot of them were raptors. We really didn't get too many reports of dead waterfowl. But it's likely that those raptors were scavenging on some waterfowl that died somewhere that nobody saw or reported. And then in the late summer, we really didn't see too many cases until the winter of 2022. We started to get more reports. And a lot of the avian influenza we found in the autumn and winter was actually by doing surveying hunter harvested waterfowl, which was something that the DNR and the USDA Wildlife Services teamed up on to monitor those birds. And then 2023, it was pretty calm. We only had a few reports, maybe, 5 geese here, 10 swans here. But it really wasn't anything particularly dramatic. And most of the detections we were getting were from those hunter harvested waterfowl, which were all ducks that, seemed really healthy at the time that the hunters shot them. And so we weren't really seeing a whole lot of mortality in the wildlife last year. Next slide, please. And the same was true for most of 2024, all the way up until December. And then December, everything caught on fire. Reports began to pick up. I think it was December 4th or December 7th, we got a report of 1,000 snow geese that died in Gibson County. Again, snow geese flock in these massive flocks, so it wasn't surprising to see a fair amount of snow geese die in that area. Another case a few days later, we had a report of about 200 snow geese that died in Vermilion County. It kind of made sense. I don't have any genetic data to back this up, but just the way it lined up, it looked like the snow geese brought the virus down, just because our earliest detections were in snow geese right along the Wabash River, which is where the snow geese like to migrate when they come down each winter. Next slide, please. And so later into January, later into December and into early January, we really started to see an increase in Canada goose mortalities and detections. Canada geese really like to hang out in urban areas. It's a lot safer for them. A lot of people feed them. There are less predators. There's usually open water. They really like urban areas. They like to scare kids, and there are more kids in urban areas. So Canada geese love urban areas. So we were really seeing a lot of detections in those high population areas. Part of that's because there's higher visibility, but again, most of these Canada geese really like to spend time in those areas. And then in early January, we got down in Jackson County. There's a group of sandhill cranes that wintered in Jackson County. Wintered meaning they didn't fly south for migration. They just stuck around in Jackson County over the winter. And so in early January, I got a couple reports of 2 or 3 sandhill cranes that were displaying signs of avian influenza, including they were shaking their head a lot. They were losing control of their limbs. They had this really bright green diarrhea, and those are all pretty good signs of avian influenza. I went down and did a site visit, and of course there were thousands of cranes down there. But the cranes that were being affected, it was kind of localized at that time. And those cranes were having a really high interaction with a few Canada geese that were in that area, but none of the Canada geese were acting sick. It was just the cranes. I thought that was really interesting. I don't have any ability to back this idea up, but I thought it might be that those geese had exposure in the past, and they had built up an immunity to it, but they were still able to shed the virus into the sandhills. I don't have any data to back that up, but it was just a thought that I had. And then later into January, those mortalities started to pick up. As of last week, there were about 600 cranes that were reported to me. That was our estimate of the number of cranes that died in Jackson County. That's most likely an underestimate because we were going off of the number of cranes that were reported. If you could give me the next slide, please. So here's a better map illustrating that. So there were quite a few sandhill crane deaths in Jackson County. And that was the case, they were kind of localized to that area up until early February. And early February is when all of the sandhill cranes started to move north. And so we started to get thousands and thousands and thousands of sandhill cranes coming up from Tennessee and Kentucky and Alabama, and they were moving across the state. And a lot of the cranes that were in Jackson County that survived, they also started to fly north and west. And so we started to get more sandhill crane mortalities in different locations. I was doing testing wherever I could, and pretty much all of the sandhill cranes that I tested were positive for HPAI. And we started to get some relatively large mortalities in Greene County, Stark County, Jackson County, Newton County. This map was made last Friday, so it's slightly out of date, but we've also had quite a few deaths in LaPorte County. And there's this tiny little county, I don't know if you can see it on the screen, Union County on the eastern border of the state. It's a little square, slightly south of the middle. There were about 50 sandhill cranes that died there on a DNR property, and I called the manager yesterday, and he said, as of yesterday, there were about 300 that have died there. So this map is a little out of date, but it does show kind of the pattern of where we're seeing mortalities in the sandhills. Next slide, please. Raptors are also pretty susceptible to the virus. A lot of these raptors are picking up the virus from, most likely from scavenging on dead birds. There are a lot of carcasses spread across the landscape, and this was a really difficult decision for me to make. It wasn't super difficult because I don't think I would have been allowed to make this decision. But we decided to not try and do carcass removal just because of the operation that would be. It would be such a huge undertaking to remove all of these carcasses off the landscape. I would have liked to do that because it would have reduced the risk to raptors, but it's not feasible. And removing all the carcasses off the landscape would not remove the virus off the landscape. And so it would be a lot of effort just to have more carcasses show up later. But raptors are able to get the virus, and we have detected it in multiple different species, including an eagle, a great horned owl, and a few different hawk species like red tails and red shouldered hawks. Next slide, please. This is a map of the total number of birds as of last Friday across the state. It's a little bit biased just because there was that one massive mortality in snow geese. But if you can see the numbers on there, you can kind of get an idea of how many birds are being affected by the virus in the wild. Next slide, please. And I can send that map to anybody who's interested, I think. What do you do if you find a dead bird? This is a question I get a lot from folks because there are a lot of people who are just finding a dead bird in their backyard, and they get really concerned about it because avian influenza is a zoonotic disease. And so we've been telling folks, report it online at on.in.gov/sickwildlife. That goes to a reporting tool. Us health biologists, there's 3 of us, we monitor those reports every day. If you report something on there, I'm getting so many reports at the moment, there's a very good chance I'm not going to follow up with you, but it does help me keep track of what's happening, where the virus is moving across the state, and what species are being affected. We're asking people to avoid contact with the carcass just because we don't want them to have that interaction. But I know a lot of people, they are really uncomfortable with having a dead bird in their yard, especially if they have kids or pets or something like that. And so we've been telling people, if it's absolutely necessary for you to remove the bird, you can double bag it and put it in the trash, as long as you wear gloves and a mask to do so and wash your hands immediately after. And if you have domestic animals, try and disinfect your clothes and your shoes before interacting with them. And you can use either alcohol or like a 10% bleach solution to do that disinfection. Next slide, please. Another question I get a lot, should I take my bird feeder down? We're not recommending people take their bird feeder down. The idea was that if these birds at the bird feeder aren't interacting with each other, then they would be less likely to spread the virus. But at this point, the virus is so established across the landscape of Indiana, taking bird feeders down really wouldn't make much of a difference for the spread. It's pretty well blanketing the state at the moment. So next slide. This was a question I got a little bit more during hunting season. Now that hunting season is over, it's a little bit less concerned, but it was a question that was given to me to answer in this topic. When you're waterfowl hunting, we're asking hunters not to harvest obviously sick or already dead waterfowl. For waterfowl that they harvest, we're asking them to process those birds out of doors so they're not inside of a enclosed space while they're cutting open the bird and potentially aerosolizing the virus. We also asked hunters to wear gloves and a mask when they're processing those birds, just to reduce the risk of contact, and to disinfect any equipment they used, any knives, tables, anything like that. And they can disinfect that with alcohol or bleach. And then we're asking waterfowl hunters to cook the meat to an internal temperature of 165 degrees. A lot of waterfowl hunters do not like to do that. When you cook the meat that high of a temperature, it makes it kind of tough, and they prefer to eat their ducks and geese a little bit more medium rare, but it's really considerably safer to cook it to 165 degrees. Next slide. And this was another question I got. This one was a little bit more difficult for me to answer. There's some question on interactions with dogs and wild birds. I did find a study that was conducted a couple of years ago on hunting dogs. And so these hunting dogs, they have a much higher interaction with waterfowl than most dogs do, because they're actually, after a hunter shoots a duck, the dog jumps into the water where all the ducks are and swims out and then picks up the duck in their mouth. And the duck is already bleeding from the gunshot, and then they bring the duck back to the hunter. So these dogs have a incredibly high interaction with waterfowl. And so a study was conducted to see how dogs react to the virus. And according to this study, a lot of these hunting dogs that they tested had antibodies to the virus, but none of the dogs showed any signs of illness, which suggests that the dogs can get the virus, but they don't really get sick from it. I don't know if that's still the case. That study was done a couple of years ago. I'm really not sure what it's like for dogs at the moment. As far as I know, it's still not a very serious virus. They might be able to carry it, but they don't really get sick from it. That's as far as I know, but some domestic animal veterinarians might be a better source to ask on that. And I think that's it. I have my email on my next slide, so if you guys have any questions that don't get answered later, you can shoot me an email. So thank you very much. I appreciate the time.
Great. Thank you so much, Eli. That was a lot of great information. I know we have a lot of questions coming in the chat, and we will answer all questions at the end. So keep those questions coming, and we will get to as many as we possibly can in our time. Again, if you think of any questions, just keep on typing them in as we move to Dr. Brown and Eric Hawkins. So I'd like to introduce our next speakers. Eric Hawkins is the state epidemiologist with the Indiana Department of Health and has been with the Department of Health since 2016. During that time, he has served as the Tuberculosis and Refugee Health Division Director, the Deputy State Epidemiologist, and as of November 2022, the State Epidemiologist and an Assistant Commissioner. Prior to arriving in Indiana, Eric worked in Arizona for over 7 years, where he worked for the Maricopa County Environmental Services Department, as well as the Arizona Department of Health Services. And Dr. Jeb Brown is also joining us as a speaker today. She is the Indiana State Public Health Veterinarian. Dr. Brown has nearly 20 years of public health experience at state, local, and federal agencies, and 3 years of clinical experience at a small animal hospital. She is board certified by the American College of Veterinary Preventive Medicine and is the immediate past president of the National Association of State Public Health Veterinarians. Her interests include zoonotic and vector-borne diseases, emerging infectious diseases, and animal-related injuries. So with that, go ahead and take it away, whoever is going to start, Eric or Dr. Brown. I think you're muted.
I'm sorry. I thought I was
No
all ready.
worries. No worries at all.
Couldn't find the unmute button. So now can you see my slides again?
Yes.
Okay, great.
Perfect. Thank you.
Thank you so much for that introduction. And Eric Hawkins and I work at the Indiana Department of Health. So our role in preparation and response for avian influenza is the human health aspects of the response. But as I'm sure you've gathered, the human health aspects of the response are highly related to the aspects of the response in wild animals and in domestic animals. So I debated, we don't have very much time with you today, so I debated whether I should leave these slides in, but I decided to leave them in because I think it's worth a review of the public health impact of influenza or flu. I think there's probably not very many people joining us today who haven't had the flu at one point or another in the course of their lives. And so flu is familiar. And I think that a familiar hazard sometimes gets dismissed or doesn't get as much attention as an unfamiliar hazard. But flu is definitely a very important ongoing cause of illness and death in people, both in the United States and in the rest of the world. A study that looked at this over a 10-year period of time in the United States found that on average during a typical flu season, 8% of the population became symptomatic, which could be as many as 41 million people. And of those, usually around 200,000 people are hospitalized, and usually around 36,000 people will have fatal cases of flu, although in a bad flu season, it can be much higher than that. So even though most of us have had the flu before, that doesn't mean that it's not a serious public health threat. Now, anyone who has had the flu before will remember what it felt like. It causes very sudden onset of respiratory and other systemic signs and symptoms, usually fever, cough, and a sore throat, but you can also have stuffy nose, muscle aches, headache, fatigue, or GI symptoms. But in some people, they're going to have flu much worse, anything from advanced upper respiratory infections or pneumonia to infection and inflammation in other organs like the heart or the brain or the muscles, can cause multi-organ failure, widespread bacterial infection as a secondary complication, or can exacerbate any underlying medical conditions. And the people who are at greatest risk for life-threatening complications of flu are people who are younger than 5 years old, older than 65, people who are pregnant, or people who have underlying medical conditions like asthma or diabetes or heart disease or other chronic medical conditions. And as hopefully we all know, flu is transmitted via respiratory droplets, and the virus that's present in these droplets is somewhat resistant to environmental influences. So if this guy sneezes on a doorknob or wipes his nose and then uses a doorknob, there's going to be viable virus left behind to hitch a ride with the next person to touch that doorknob. And this is why flu is so contagious. Now, I debated if I should take these slides out, but I thought, and Eli went over a couple of these points, but I think it's helpful to sort of do a little refresher on basic flu biology. And this might also help you all listening out there be savvy media consumers as you're reading stories about flu, because it is a pretty big complex of viruses. There's actually 4 different types of flu, but only type A and type B are important to public health. And of type A and type B, only type A is the group of viruses that can also infect animals. Type A also has a tendency overall to cause more severe illness in people than type B, although type B certainly is capable of causing severe illness in people. And type A is also capable of major genetic changes. And this is sort of the core of our concern about flu as a potential cause of a pandemic. Eli went over a couple of these things too. He talked to you about how flu viruses or flu A viruses are categorized according to these surface proteins, hemagglutinin and neuraminidase. And these are shown as little blue blobs and little red blobs on the surface of this flu virus in this diagram. And I think he went over that pretty well. I wanted to add that inside this virus is the genetic material. And flu viruses have RNA as their genetic material, not DNA. And that's important because it's a lot easier to make mistakes when RNA is making copies of itself versus DNA. DNA is sort of, it's a double-stranded piece of genetic material. It's harder for mistakes to be incorporated into the progeny viruses when the virus is replicating. But if it's RNA, it's much easier for mistakes in copying that genetic material to be passed along. The other thing about the genetic material of a flu A virus is it's segmented. It's broken into 8 different pieces. And those pieces actually have the ability to combine together in certain circumstances to create new viruses. And we'll talk more about that here in a little bit. As Eli shared, some flu A viruses are endemic in wild freshwater waterfowl. Almost all flu viruses are capable of infecting birds, flu A viruses, but the avian ones are simply those that we often find associated with wild waterfowl. And as Eli told you, they're classified based on their effects on chickens. And that's an important point because avian flu viruses are also capable of causing illness and death in people, and both highly pathogenic avian influenza strains and low pathogenicity avian influenza are capable of causing human illness. So the highly pathogenic moniker is really just related to chickens. It doesn't have any correlation with its ability to cause severe illness in people. So in people, when we see avian influenza virus infection, it's usually rare, it's usually sporadic or kind of random, and it's usually in people who have close contact with infected animals. We don't usually see ongoing human-to-human transmission from a person who's infected with an avian flu A strain. There's 3 main types of avian flu A viruses that we've seen be associated with human illness: H5, H7, and H9. And again, remember, this is referring to one of those surface proteins that Eli told you about. Compare that to seasonal flu viruses, are most often H1s or H3s. So which is actually helpful when we are doing laboratory testing, because by looking to see what type of H protein a flu virus has, we can actually tell if a person had seasonal flu that they probably caught from another person, or if they have one of these subtypes that's more often associated with birds. In people, there can be a spectrum of illness when infected with one of these avian flu viruses. It can be anything from just conjunctivitis or pink eye and no other symptoms to the flu, as we all understand flu-like illness. And just like other flu viruses, these viruses can cause severe respiratory illness or multi-organ failure or even death in people. And there is a potential for avian flu viruses to undergo changes that would allow them to be more easily transmitted from one person to another person. And if they acquire the ability to do that, and they acquire the ability to cause severe illness in people, that's the recipe for a pandemic flu virus strain. And that's the type of event that is thought to have given rise to the global flu pandemic of 1918. Now, you're not meant to read everything on this graphic. The main thing I wanted to talk to you about on this graphic is that because they're RNA viruses, that's easy to make mistakes in replication of the genetic material, and they have these segmented genomes. Flu A viruses are always changing. They're always changing a little bit over time due to these random mutations in the RNA that are then passed along. And this is why when the vaccine manufacturers make flu shots, they have to change what formulation they're putting in the flu shot every single year, because the flu virus that's circulating next year, even if it's the same H type and N type, it probably has enough mutations in it that it's different, and the flu shot from last year won't cover it anymore. So that's one thing. But the other thing that can happen is that passing through other hosts, non-bird hosts, can sort of result in the selection of flu viruses that have undergone more major changes. And the thing that I think public health officials are most worried about is this phenomenon of reassortment. And this is what might happen if an avian flu virus infected an intermediate host, like a pig, or it could be a person, and a human seasonal flu virus infected the same host, whether it's a pig or whether it's a person. And then you have two completely different flu viruses in the same mammalian cell. And when those two flu viruses go through the process of replication, remember I said that the RNA is broken into 8 different pieces. All of those pieces get jumbled together. And the progeny virus that is the result of replication could look completely different from either of the two parent viruses. And what that means is that a novel strain of flu could emerge as a result of that event that resulted in the combination and mixing of genetic material from two different viruses. A novel strain could be very bad, because if a novel strain of flu is able to make people very sick, and if a novel strain of flu is able to be passed easily from one person to another, and it's a novel strain, that means that people, the general human population, might not have a lot of background immunity to this new virus. That's the recipe for a possible pandemic. So when we're talking about what's going on with avian flu right now in wild birds and in domestic animals, and talking about what's going on with people, this is the thing that I think public health officials are most worried about. So I'm going to hand it over now to Eric Hawkins to talk to you a little bit about what we've been seeing lately in terms of human epidemiology.
All right, thanks, Jen. Can you hear me okay?
Yes.
Yeah, perfect. So thanks, everybody. Thanks, Jen, for that awesome background. Also, Eli, for your very thorough presentation as well. And I think some of this, so this first slide here and these next few slides are going to go through sort of that timeline of where we've seen HPAI, or influenza A, H5N1 in the United States. And I think shortly before, I believe what Eli said in Indiana was March of 22, where we saw it in wild birds. So not too long before that, nationally, we were seeing it in January. And then into February is when we started to see it in our in poultry. So then I think the next step here is to think about is like what happens after that. Are we seeing human infection? So we did see in the US our first human infection in 2022. This happened in Colorado, and this person had exposure to poultry. I think there's actually some thoughts that this person was actually, their nasal passage was actually contaminated by the environment that they were in. So nonetheless, this was considered the first human infection in the US. Now, fast forward to last year, early last year, 2024, is when you started hearing about Texas and we have dairy cattle that are now infected with H5. So that occurred, like I said, down in Texas, and quickly sort of rolled out after that. I think during that time is also when you start seeing sort of our human surveillance side of things start to pick up. We're more sensitive to these things. We're seeing a new species or a new mammal that now has been infected with H5. So we become more sensitive to that, and I think that's a good thing in a sense of that's how we do our surveillance. We try to see what's happening. But what we saw after that, shortly after that, was that our number of human cases drastically changed. So we started seeing a lot more human infections following that. Once it moved into the dairy cattle, and then in September of last year is also marked another point, and I think it's an important point, that there was the first human case where there was no known animal exposure. So they didn't have any exposure to poultry farms or a backyard flock or dairy cattle. They actually, it's possible they picked up in the environment, but this also just happens when we're talking about viruses. People can pick it up. It could have been an incidental pickup just in the environment that they cross paths with some mammal or some bird that may have had it. And then unfortunately, early this year, I'm sure you all saw this, we did see our first fatal human case in the United States, and this occurred in somebody that in Louisiana who did have really high risk exposure. They had a backyard flock, I believe, of 6 birds. There were several dead birds, and they were actually handling these dead birds without any sort of PPE. And ultimately, that person ended up becoming infected and unfortunately passed away. If we look at some of the animal side of things, as far as what we're seeing across the country, this is from the USDA. They are doing lots of work with state animal programs. But what you can see here is that commercial flocks, there's been lots of infections within commercial flocks across the country. I think this is over time, Jen. I think you pulled this slide together for us, but if we looked at this in the past 30 days, you'd actually see that Ohio is actually really high. I think they're one of the highest states right now, just as far as activity goes with commercial flocks. Next is the backyard flock. So you can see pretty much every state, if not every state in the U.S., has had detections within backyard flocks. So I think that's important to note, especially when we're talking about human health and sort of that potential for exposure. And then similar to DNR, I know DNR has been very busy with wild bird detections and die-offs. Also, our Board of Animal Health has been extremely busy over the last couple of years, 2-3 years. As you can see from this map and some of this data in here, we've actually had 33 affected premises, so poultry operations within the state that have been infected. And that's been across 14 counties. And unfortunately, some of those counties are affected more than others and then have multiple operations within those counties. But important to note here that our animal partners have been extremely busy as well. And as you can see down there, just a quick summary of the number of poultries or birds that have been affected in Indiana. It's been almost 7 million birds since going back to 2022. Now, speaking of dairy cattle, you can see it's not quite as widespread, but we continue to see more states sort of pop on there. Some of the national calls we've been listening to, some of these detections have slowed down. A lot of the new states that have been popping on have actually been picked up through the USDA's National Bulk Milk Testing Program. And this is a program that they're working with states, with different states across the country to actually test milk supplies before they've gone through the pasteurization process. So I know at least a couple of the last states that have been picked up through this process, or that have been added to this map, I should say, have actually been picked up through that process. So again, I think every state will eventually sort of be part of that bulk milk testing program. But it's important to note that that's actually happening now. And it is helping to identify some of these infected herds. Speaking of from the CDC's perspective, and this is data from the CDC, looking at the human health side of things, we have yet to see any person-to-person spread when we're talking about H5 or avian influenza. We're still marking this as a low risk to the public. In total in the U.S., we've seen 70 human cases, and unfortunately, like I mentioned earlier, we have had that one human death. Looking at the human sort of the map of human cases, you can see that there's a majority of these have occurred in individuals that had exposure to dairy cattle that were infected, so dairy cattle herds. And then you also see a significant amount of people that have been exposed through poultry operations. I did mention that we've had a handful of cases that have no known exposure, so those are highlighted on there as well. But just to give you a good representation of where we're seeing the cases in the U.S. And Jen talked a lot about our sort of influenza in general, just sort of non-pandemic or non-H5 influenza. Across the country, I think we have a pretty good network of hospitals and laboratories that partake in our national flu surveillance, including Indiana. We have a lot of partnerships. We do testing in our state laboratory for influenza. And through that process, we've actually, in the US, we've actually picked up 6 human cases through that. And these would be individuals that have gone to a healthcare provider and had a specimen drawn, and it turns out to be H5. So I think it's important to note that we do have surveillance things in place. We do have influenza testing happening across the country to help differentiate what's happening seasonally versus are we picking up any sort of influenza H5. It's also important to note even though we haven't had a lot of human cases and very few human cases when you're talking about the amount of animals that have been infected in the US, it's important to note that our public health partners, including here in Indiana, that we are taking lots of steps when we have these exposures or we have farms that are infected. We're working with individuals that may have been infected. We're doing monitoring of those individuals, looking for symptoms. We're testing those individuals, offering testing, and we're also offering post-exposure prophylaxis if needed. So lots of work being done there. And through that process of just monitoring, we've also picked up additional cases. So sort of getting to the take-home messages here with HPAI, it is primarily at this point in time an animal health emergency, but as Jen has been sort of gave us the background on there, I mean, there's a potential for a pandemic, and that's why we as public health are actively involved, and we're taking part in this process. So the risk of the general public is considered to be low. I think where we need to focus and we continue to focus on individuals that are that have high risk exposure. So those, when we think about that, we think about those that have occupational exposure. So those are working in operations where there's sick birds, sick cattle, or other animals that may be at risk. Those are the individuals that may be becoming sick. And then it's also important to note that those that have been sick have not had severe symptoms. So I think even some of these very few have been asymptomatic. However, I think a majority of our cases actually have very, very minimal symptoms, and even conjunctivitis may be the only symptom. When we're talking about food and milk supply, I think it's really important to note that the food products that are from sick animals rarely enter into our food supply chains. And then also really important to remember, and always a take-home message for us, is that the proper cooking of meat and pasteurization of milk products actually kills and inactivates the virus itself. So as far as prevention, our messages for the general public and continue to be the same, is avoiding that direct contact with sick or dead birds or other animals. Make sure that we're not touching animals that or contaminated tissues or bodies of animals that may be infected. And then CDC recommends that we avoid consuming raw milk or milk products, especially from animals that we know that are ill. And then those that are at high risk of exposure, especially those with occupational exposures, making sure that we have proper PPE, and then there's education on that PPE. And then, as Jen alluded to, an important tool that we always have is an influenza shot. And we know that this won't protect us against H5. However, it's important to note that some of the public health risks around this is reassortment, and the concern would be that seasonal flu mixes with a pandemic flu strain or H5 strain. So those are the basic take-home messages that we like to pass along. And Jen, I don't know if you want to touch on this. I know this is your team that primarily put this together, but some of the resources that you guys have created.
Just bookmark our webpage if you'd like more information from the human health perspective. I think all of our 3 agencies, the Board of Animal Health, the Indiana Department of Health, and the Department of Natural Resources, I'm pretty sure we all have agency avian influenza pages, and you can get up-to-date information from each of our agency perspectives if you seek those pages out and bookmark them. So I would encourage you to do that.
Great. Thank you so much for your presentation. Thank all 3 of you for all of this fantastic, timely information. Again, as we're starting to go through questions, go ahead and continue adding questions to the chat box. And I'm going to start going through everything. Really quick before I do, just wanted to catch anyone before you pop off. Continuing education certificates for DVMs and RVTs will be sent out in the next week or so. So just wanted to throw that out there. And again, any questions that are not addressed during this webinar, because I know we only have a few minutes, will be compiled, answered, and attached to the YouTube posting in approximately 1 week on the BOA YouTube page. So just a couple of questions that I think we could get through. So for Eli, how much of a role do sparrows have in the spread of the virus? Can you comment on the roles of passerine or songbirds carrying the virus?
Yeah, songbirds can definitely carry the virus, and they don't really get sick from it. And so it's really hard to tell where the songbirds are carrying the virus because we don't have mortality events in them. Usually if a songbird has the virus and they get sick, it's because they have another comorbidity with it. So it's kind of hard to track where the songbirds have it, but at this point, we're just kind of assuming that it's blanketed across the landscape, the virus is. And so songbirds, they can carry it, but we don't know to what degree. I wouldn't mind doing a study on the prevalence of avian influenza in songbirds, but that would take a lot of time and money, and I don't know if we have that.
Fair enough. What is the strategy of the DNR for testing wild birds and mammals for HPAI? You covered it, but just wanted to reiterate because it was a question.
Yeah, so we are testing wild birds wherever we had a large mortality event. Or even smaller mortality events. And we were trying to locate the virus in new areas. So whenever we had a mortality event in a county where we had not already detected the virus, we were trying to do some testing there just to kind of show geographically where the virus is at in the state. And also if we have different species, I think it's interesting and good knowledge to have what species are being affected by the virus. For example, we had a red-throated loon, which are very rare in this state. But we had a red-throated loon die, and we got that one tested, and that was actually negative for avian influenza. They're still doing a necropsy to figure out what that was. But we do try to test new species when we have those mortalities. And we're trying to detect it in new counties as well.
Thank you. One question, are those birds that have been found deceased and reported online just considered positive with no testing to confirm?
Yeah, a lot of that depends. If it's a species where a species that we have detected the virus in, and it's in an area where we have detected the virus, we're assuming that the species has the virus. I have a limited diagnostic budget, so I'm trying to be strategic where we do our testing. So we can't test every bird. So we just try to follow that strategy of testing new locations and new species.
Thank you. When cleaning a bird feeder or bird bath, should you follow the same PPE guidelines as you would if you found a dead bird? I'm thinking gloves, mask, that kind of thing is part of the question.
Yeah, even outside of avian influenza, it's a good idea to wear gloves and a mask when you're cleaning your bird feeders because songbirds can carry salmonella with them as well. So it's always a good idea to wear gloves and a mask when you're cleaning your bird feeder, just in case.
Great, thank you. Have you come across any white-tailed deer with the virus, and any specific recommendations for hunters?
For white-tailed deer, I don't think anybody's detected the virus in white-tailed deer. I'm not sure if the USDA Wildlife Services has been doing testing on those. They do a lot of testing on white-tailed deer across the country. And as far as I know, nobody's found it in white-tailed deer. I don't think we've had any detections. We certainly haven't had any in Indiana, but even nationally, I don't think there have been any detections.
Great, thank you. Has the virus been detected in any wild turkeys, and are there any precautions we should do going into spring wild turkey season?
In Indiana, we have not detected it in wild turkeys. I want to say it's Wyoming and some of the western states they found it in wild turkeys, but in Indiana, we haven't found it in wild turkeys. They don't usually, historically, they don't seem to be as affected by avian influenza as some of the other taxa like waterfowl or raptors.
Great, thanks. Which is interesting because domesticated turkeys are very affected. How long does the virus live in the soil where the dead carcasses are being left?
I've been trying to learn that information, and different sources have different ideas. And so it largely depends on the temperature. So the virus will stay stable in the environment longer at colder temperatures. And as the temperatures start to rise, the virus will not stay stable in the environment as long. So I think I want to say around 60, 70 degrees, it might last maybe 2 weeks. At like sub-freezing temperatures, I've read it can last up to 2 months, and I've also read that it can last indefinitely. So there's a little bit of debate going on there. But as the temperatures start to warm up, it doesn't seem to last in the environment quite as long.
Okay, thank you. One more question for you. If it's been found in other mammals like foxes, should we assume it is in squirrels and rabbits as well?
Not necessarily. Different species react differently to the virus. So sometimes there's some species that aren't going to be affected by it at all. It's certainly possible. I know it's been detected in some field mice and stuff like that before, but I don't think we've detected it in any squirrels that I know of. And I want to say we've never detected it in cottontail, eastern cottontails, which are the rabbits that we have here in Indiana. But I think there might be one rabbit species where it's been detected in, but that was super rare.
Okay, great. Thank you. All right, continue. I know we are a couple minutes past time. We have a couple questions for Dr. Brown and Eric before we finish out. Again, if we don't get to your questions, we will compile them all and have them on YouTube. So for Dr. Brown and Eric, what % of human influenza testing is looking for H5N1? I think, Eric, you briefly talked about this. The requested test, is it regularly performed?
Yeah, so Jen, please step in if you need to add to this or have anything to add. But so it's important to note that avian flu H5N1 is a influenza A. So the routine tests that we see in our even some of the like over-the-counter tests, like those rapid tests, they pick up influenza A or B. Same thing happens when you go to your doctor's office. It'll pick up influenza A or B. So if an individual had influenza A H5N1, it would be positive on a flu A test. at that point is where some different steps need to take place. So some hospitals, some laboratories have the ability to say it's influenza A, but it's maybe it's H5, or they may say it's untypable, and then it would require additional testing. So that's when the epidemiology comes into play as well. It's like, does that individual have risk factors? Have they worked with sick animals? Should it be forwarded on to our state public health laboratory at Indiana Department of Health for additional testing, and then that's where we would do additional testing, and then ultimately send it to CDC for final testing. So to that end, to simplify it, I think our typical influenza seasonal, whether it's seasonal or avian H5N1, those tests will pick it up and it will be positive on those. It's the additional tests after that take place. Hopefully that helps.
Great. Yeah, thank you. I appreciate that. One question, if people do not get a yearly flu vaccine, does it increase the chances of HPAI mutating with influenza A or B, potentially making it more contagious and dangerous in humans? Touched a little bit on this, but specifically regarding the vaccine.
Yeah, so seasonal influenza vaccination is always, I mean, that's one of our core public health sort of preventive efforts is influenza vaccination. So I think when we look at the context of what's occurring and we look at H5, there's still low risk. The human risk is pretty low. So I think it's an important message here. We have respiratory season. There's lots of things that we can do to prevent spread of respiratory illnesses, including one of those tools is actually the influenza vaccine. So in that sense, I think the message here is the seasonal flu vaccine is perfect for, not perfect, but it's a good tool that we need to use for seasonal influenza. I think in the messaging and where Dr. Brown and I both sort of talked about this slightly is that especially those individuals that have potential high risk exposure to animals that are infected or working closely with animals that are infected, I think the seasonal flu vaccine is one of those. It's another tool there that could potentially help reduce that sort of reassortment even more so. So I think I just go back to, I think seasonal flu is really important right now for seasonal influenza, not necessarily H5 right now. Jen, did you have anything to add to that? Maybe we lost her.
Yeah, might have.
No, I'm here. No, sorry, I
there.
was shaking my head. No, and I
Oh, okay, perfect.
didn't realize you couldn't see me. No, I don't have anything to add.
No worries. I think we'll wrap up with one final question as we're getting to almost 10 after 1. What type of surveillance is being done in swine and swine workers? I know we talked briefly about swine, maybe also broaden it to avian and avian workers.
Yeah, I can take that one. So we're not targeting swine workers for additional enhanced surveillance at this time, because I am not aware of any epi evidence that would suggest that that there's active circulation of this virus among commercial swine or wild swine for that matter. However, that doesn't mean that surveillance isn't occurring in swine workers, because those other surveillance activities that Eric just described are happening for everyone. So if someone develops a flu-like illness and has a positive rapid test for flu A at urgent care or in the ER, then we're encouraging subtyping, additional testing to be done to determine whether that person has seasonal flu or whether they have something else. So if someone went to, say, an ER and had a positive flu test and the hospital lab tested it and found this isn't H1 and it's not H3, so this doesn't, they're flu positive, but it doesn't seem like it's a seasonal flu, then we've been asking hospitals to send those specimens to us at the State Health Department lab for additional subtyping. So that's how we would catch that person. And so in poultry workers, we're doing additional active surveillance, because currently our understanding is that poultry workers and dairy workers are at the highest risk of exposure. If our information changes and we learn that we have reason to be concerned about circulation in swine workers, we will certainly add them into the mix for our enhanced surveillance. But currently, for dairy workers who are exposed to confirmed or suspected positives, or poultry workers who are exposed to confirmed or suspected positives, the first thing that we're doing is we're allowing our colleagues at the Board of Animal Health to handle the animal emergency aspects of the situation, and then working with them to get contact information for folks who've been exposed, so we can reach out, see if they've been symptomatic, offer them testing if they want it, offer them Tamiflu if they want it, just in case, and then helping them understand what actions we're requesting if they become ill. Because if someone who has been exposed to infected animals becomes sick, we want them to be able to get tested right away so that we can see if they have H5 avian influenza or if they have something else. So we are very much actively engaging with people who we know have been exposed to infected animals to help them get what they need.
Great. Thank you so much. Thank you, all three of you, for a fantastic presentation. Thank you for answering all of these questions. And since we are 10 minutes after the hour, I will go ahead and let you all go to your day. Thank you all so much for joining the presentation today. And we look forward to sharing this webinar with all of you on our YouTube channel, the Board of Animal Health's YouTube channel. And I hope you have a wonderful day. Thank you.
Thank you much.
Thank you.